When they're not nearly as talked about as the nucleus or the mitochondria, ribosomes are easily one of the most important organelles in the cell. There are about 10,000 per cell, so you have about 4 quintillion in your body at any given time. Ribosomes are synthesized in the nucleolus and can be found in two major places in the cell. The first place is the rough endoplasmic reticulum. It's studded with ribosomes and tends to make proteins that attach to or leave through the cell membrane. The second place is the cytoplasm. These ribosomes are free-floating and usually make proteins that are used inside the cell. Ribosomes are a little more complicated than they look, but they're still fairly simple compared to most organelles. They're made up of two subunits, conveniently called the large and small subunits, and they have three internal tRNA docking sites, called the A site, the P site, and the E site. Like I said before, ribosomes synthesize proteins, and they do this through translation. To translate mRNA, the ribosome binds to an mRNA strand and reads it in a three-nucleotide sequence called a codon. There are 64 different codon combinations, which code for 20 different amino acids. This is a typical amino acid table, but you don't really have to worry about memorizing it. You'll almost always be provided with one. But the important codons that you should remember are the stop and start codons. Stop codons tell the ribosome to stop adding amino acids to the protein chain, but they don't actually code for any specific amino acids. Start codons, on the other hand, code for the amino acid methionine, and they initiate translation. There's only one start codon, so every protein starts with methionine. Now that we understand what codons are, let's take a quick look at how translation works. Translation has 6 major steps, so we'll go through them one by one. The first step is binding to the mRNA strand. The A site always binds to the start codon, and the nucleotides face the large subunit. The second step is for the ribosome to grab a free-floating tRNA molecule. tRNA molecules consist of an amino acid and an anticodon. The anticodon dictates which codon it binds to, and the amino acid is donated at the P site. We'll look at that in a sec. The third step is for the anticodon to line up with the start codon as the tRNA moves into the A site. The fourth step is for the ribosome to grab another tRNA molecule complementary to the next codon in the strand and move it into the A site. At the same time, the tRNA that was in the A site moves to the P site. The fifth step is for the tRNA in the P site to move to the E site and donate its amino acid to the next tRNA as it moves from the A site to the P site. That probably sounds a little complicated, but basically, the tRNA molecule moves from the A site to the P site to the E site, and when they reach the E site, they donate their amino acid to the tRNA in the P site. These 5 steps repeat, and ribosomes continue stringing amino acids together until it reaches the stop codon. The stop codon initiates the 6th and final step, which is for ribosomes to release both the mRNA strand and the protein. The protein folds by itself, and the ribosome is free to bind to new mRNA strands. We just went over a lot of content really quickly, but here's a brief summary of how ribosomes work. 1. Ribosomes read 3 nucleotide sequences called codons. 2. For each codon, a ribosome summons a free-floating tRNA molecule with a corresponding anticodon. 3. Ribosomes take the amino acid off tRNA molecules and string them together to make proteins. And 4. Ribosomes aren't involved in the protein folding process.